Welcome back, everyone. Next, we have Coeptis Therapeutics, trades on the NASDAQ under the symbol COEP, and it's a biopharmaceutical company developing innovative cell therapy platforms for patients with cancer. Please welcome President and CEO Dave Mehalick. Nice to see you, Dave.
Nice to see you too, Anna. Thanks for having me here today.
All right, the floor is yours, and call me back when you're ready for questions.
Okay, sounds great. Thanks. And thanks everybody for taking the time to listen to information about our company today. As Anna said, my name is Dave Mehalick. I'm the president, CEO, and one of the founders of Coeptis Therapeutics. Coeptis trades on the NASDAQ under symbol COEP. I'll leave up for a moment the cautionary forward-looking statements and the disclaimers. And I'm sure everyone has had full time to read all that during that period. So as Anna said, we're a Pittsburgh-based clinical stage biotech company focusing on innovative cell therapies, mostly for use in oncology and other diseases. I'm going to go over some of the company's general highlights. We do have 3 core technologies, which is quite a bit for a company of our size. And I'll speak to each of these individually as we move on. The first being our universal SnapCar platform, the second being our gear engineered cell therapy, and the last being our cord blood derived allogeneic cell generation platform. And each of these technologies are platform-based technologies. We have 2 clinical studies currently going on, one for relapse refractory AML and high-risk MDS patients, and the other one, which has closed out enrollment and study, was for subjects hospitalized with COVID-19, although I'll get to that a little bit more. We've been public on the NASDAQ for over 1 year, as I said, under the stock symbol COEP. We have currently 5 development and clinical collaborations, them being the University of Pittsburgh, the Karolinska Institute out of Stockholm, Sweden. We work with the Fred Hutch Cancer Center in Seattle, Duke Cancer Institute, and Novant Health. And we do have over 13,000 square feet of research and development capabilities, mostly out of our research lab in Seattle. These are some of the corporate milestones over the past couple of years. You can see in 2021, we secured the rights to gear from Vigent Bio, which was a company that was pulled out of technology coming out of some of the best minds out of Karolinska. In 2022, we licensed the exclusive license for SnapCar from the University of Pittsburgh, and later that year we listed on the NASDAQ. And in 2023 was another banner year for us. We did license the clinical programs from Devera Therapeutics and the cell generation platform. We were able to expand the SnapCar license with the University of Pittsburgh to include NK cells as the infector cells, utilizing with the CAR technology that I'll explain. We really onboarded the R&D site in Seattle, where we're doing most of our research right now. And we are going to close out the clinical trials that we're in. Let's go to our pipeline. We have a pretty diverse pipeline, especially, again, for a company of our size. We have the 2 trials I spoke of that are currently ongoing. We have preclinical work going on with the CD38 gear platform, and we have preclinical work going on with SnapCar for 3 different areas. And really, the company has abilities to utilize allogeneic cells due to our acquisition of that cell generation platform from Devera, and we also do some work on the autologous side as well. Let's talk a little bit initially about our most frequent acquisition, which is the proprietary allogeneic immune effector cell generation platform that we acquired from Devera. And this is a very key component to our company, and really allows us to use our engineering platforms with our Snap and Gear in a truly off-the-shelf fashion with allogeneic cells, effector cells, that are being created through the use of FDA-regulated cord blood as a source material. And that's a really big point for us, because it's allowing us, through use of the cord blood in a pool donor scenario, it's allowing us to be able to utilize with patients without requiring HLA matching. So we do not need to match the blood and the cells with the patient, as it can the cells that we're creating can be truly off the shelf and be used in anybody. As you'll see on there, this is a completed manufacturing and cryopreservation process. It has been put into patients already, and it has been seen by the FDA. And we have approved INDs for this already that we've been using it for. It's reproducible, it's scalable, and very important, it has extremely low cost of goods, which is something that you do not see at all in cell therapy. And I think this is going to come back to Coeptis's desire. And one of our mission statements is to universalize cell therapy. And what we mean by that is being able to make it used for the masses at early stages in their diagnosis instead of much later stages, because insurance won't pay the high prices of the current therapies that are out there. Let's move on from that. We will talk about the 2 clinical trials we have. The first one on the left, really that was for COVID pneumonia patients hospitalized. And we were able to, you'll see, we were able to enroll, we were able to dose at the highest level, our pooled donor cells. And that had never been done before. We are the first company that we know of that has been able to dose pooled donor, no HLA matching, and have been able to dose with no dose limiting toxicities, and all dose levels were fully tolerated. So that's extremely exciting news for us. Speaking a little bit about that trial, we are looking to push that trial forward now and augment it, or I would say even modify it, to be able to be used in patients that have viral or rather respiratory infections due to viral. So it wouldn't just be COVID, it would be hospitalized respiratory infections due to viral issues. Our second indication is for relapse refractory AML and high risk MDS. We have 2 more patients to be fully enrolled, and we expect to be fully enrolled before the end of the year. And again, maximum dose levels with no DLTs in this as well. And we do expect data on both of these to be released sometime in the first quarter. Let's talk a little bit about SnapCar. SnapCar is a technology that we licensed and acquired from the University of Pittsburgh. Extremely exciting technology with extremely wide reach of opportunity. And what we have created there with SnapCar is we've created a universal CAR. So the universal CAR can be utilized with any type of effector cell and then be put into a patient's body. And then what we do, and you'll see on the left, that visual on the left there, it shows that what we do is we create that SnapCar cell and then we tag a monoclonal antibody that basically connects to the SnapCar cell and then attacks the tumor. So what that does is it allows us to utilize the same cell and attack many different cancers and attack the cancers all at different target points on the cancer, which is extremely exciting. It allows us to basically titrate or dose the amount of CAR cells that we are giving to a patient. So it possibly could allow us to avoid what sometimes people get in these CAR therapies as they get cytokine release storm, which causes a lot of issues. It allows us to possibly avoid that. But really exciting areas. Again, it allows us to attack the cancers at different target points, which therefore we feel will allow us to be much more effective in not only attacking the cancer, but also prevent the cancer's innate ability to mutate and evolve away from how it is being attacked. This is a treatment approach. We would have a patient that would present with cancer. We would be able to utilize that one universal cell, infuse the patient with the one universal cell when they come in, as opposed to the current strategies, which are to have a patient go into the hospital, pull cells from the patient, which of course is an autologous setting, engineer the cells, expand the cells, put them back in the patient's body 25 to 30 days later. Very expensive procedure. In our procedure, we have the cells ready to go. They are thawed out, they are put into the patient, and then the oncologist will be able to select from a catalog, if you will, of anti-tumor antibodies that it could address different malignancies and different target points upon the malignancy. So very, very exciting there. This is an example of the ability to do not only what you've seen basically in the CAR T therapies out there. They've only been affected in the hematological malignancies. Due to the way that SnapCar has been constructed, we're seeing effectiveness in solid tumors as well. which opens up, a much broader area of cancer treatments that are currently being used for CAR therapies. And this gives you just a small example of some of the early opportunities that we are looking at with SnapCar. You will see on the left an effector cell, whether that be a T cell, an NK cell, a macrophage, but connecting into the CAR, being able to be put in the body. And on the right, you can see the different antibodies that you could tag and therefore go after these different areas, such as B cell malignancies, AML, multiple myeloma in the hematological malignancy area, and then solid tumors. There's a vast amount of those also that we would be able to target utilizing, again, the same universal cell. I'm going to advance now to our technology called GEAR. GEAR stands for genetically edited antibody resistant effector cells. And really what GEAR does is it is an engineering that prevents the cell fracticide, the effector cell fracticide. Effector cells, of course, being cells like natural killer cells, prevents the effector cell fracticide that occurs when you introduce a monoclonal antibody therapy to a patient. It's going to be, I'm going to show you the next page. That's going to give you a little bit more explanation of how it works. Currently, on the left, you see the current anti-CD38 pathway monoclonal antibodies, and you could see the tumor. You could see the expressed CD38 proteins on the surface of the tumor, and then those Ys that are the monoclonal antibodies that are attacking the tumor. The problem you have there is you also see the natural killer cell that will, of course, express the CD38 proteins as well. And you have the negative off-targeting effect of the current monoclonal antibody therapies that attack those natural killer cells and kill them off as well, leaving a patient with an immune system that is depressed. What we have been able to do is on the right, you would see the monoclonal antibody still doing the work it's supposed to do, attacking the tumor cell and killing them off. But we have created a effector cell that is immune privileged. Immune privileged meaning that the monoclonal antibody no longer recognizes it and is able to attach to it, and the natural killer cell remains fully intact. And this is an important point. The CD38 proteins on the natural killer cell remain fully functional. There's been a lot of other companies in this space that have been trying to accomplish this and have attained, tremendously large market caps and valuations and raised, billions of dollars doing this, but have failed because they have really looked to eliminate the CD38 protein from the surface of the cell, and therefore the cell just wasn't functional and didn't work. But we were able to show thus far that we've created an immune privileged cell that is accomplishing exactly what others have tried to do. Talk a little bit about our management team. Myself as a co-founder, president and CEO. Dr. Colleen Delaney is our chief science and medical officer, certainly one of the most recognized scientists in the area of cell generation, creating of cells, especially in the area of cord blood. Brian Cogley is our chief financial officer with years of experience in corporate finance in the science and pharmaceutical. Daniel Race is a co-founder with me. He's our VP of operations. Christine Sheehy, also a co-founder, and she's our VP of compliance. And we have Laura Silverman, who is our VP of clinical operations. Everyone with a lot of experience in the space. And lastly, I'll show you our scientific advisory board. And Dr. Evan Alici, Dr. Hans Gustav, and Dr. Wagner, all some of the most renowned scientists in the world, especially when it pertains to cell therapy and especially natural killer cells. Dr. Alici is the head of cell and gene therapy at the Karolinska Institute. For those of you that aren't familiar with Karolinska, it is world renowned for that's where they name the Nobel Prize in Medicine every year. Dr. Hans Gustav Lundgren is the former dean of research of Karolinska. He founded their infectious disease institute. He does sit on the Nobel Prize assembly to nominate the person every year. And Arnika Wagner is a professor working with Dr. Alici and is one of the co-creators of the GEAR platform. So we do have a scientific advisory board that is, world renowned and some of the top minds in this space. And I'd like to thank you for taking the time with me today, presentations, and the call back, Anna, to see if she has any questions for me.
Great job, Dave. Yes, important work you're doing here. I want you to talk a little bit more about the SnapCar. It seems to have extremely broad applications to so many different cancers. So talk a little bit more about that, and how does this affect your ability to license out this specific technology?
Yeah, that's a great question, Anna. It does. As you could see when I was going through the presentation, SnapCar, because we have the ability to create a universal cell, instead of creating a CAR construct that is a singular cell that is only good to attack a certain point on a certain cancer, we've created a universal CAR, which allows us to now utilize antibodies that are created from any company. So there is, hundreds of different antibodies that are owned by large pharma companies that gives us the ability to license with them, and gives the large pharma companies the ability to utilize these to, one, expand their IP, but dramatically expand the effectiveness of their current antibody portfolio. So the license opportunities in SnapCar are ridiculously enormous, because you're talking every major pharma company should be calling us if they haven't already.
Absolutely. Seems like it. You mentioned gears, so talk a little bit more about that. And it does seem similar nature to the therapy that failed for the Fate Therapeutics. So why do you feel Coeptis can succeed here where they didn't?
Yeah, that's a really good catch on Fate. I was trying to push it down a little bit earlier, but yeah, they were trying to do the same thing that we were doing, where they were trying to create a cell that was not being attacked by the monoclonal antibody. What they did is they eliminated the target on that cell, on the natural killer cell. And by eliminating the target, though, they did succeed in not having it be attacked by the monoclonal antibody, but the cell failed. The target, the CD38 protein, is necessary for cell function and viability. And we had recognized that. So we created a different technology that allowed that to be totally functional, but still avoided attack. So, and if you look at Fate, at one point, they were a company similar in size to us. They at one point grew to an $11-12 billion company. And kind of a lot of it was on the back of them hopefully succeeding in this area, where we feel that right now we are succeeding in. So it's very exciting for us there.
And also, the FDA recently announced a cancer safety review regarding secondary cancers that could develop due to CAR T therapies. I know we've interviewed a lot of people talking about CAR T therapies. Can you talk a little bit about that, and how does this affect Coeptis and the SnapCar that you mentioned earlier?
Sure. Yeah, that's been a big thing that's just come out over the last couple of days. So yeah, current CAR T therapy, really the only thing that's been approved has been in the autologous setting, which is the usage of a patient's own patient's own cells. And those cells tend to persist. They're engineered cells put back in a patient's body that are no longer native, that tend to persist in the patient. So the FDA is recognizing that they may cause some mutation that causes a cancer later. For Coeptis, it doesn't have any negative effect on us at all, actually, because Coeptis has created, first of all, we're not a CAR T company, we're a universal CAR company. So that CAR can be used with natural killer cells, macrophages, other effector cells that do not persist in the body later. But on top of that, we're also an allogeneic off-the-shelf therapy company. So we're not utilizing the company's own cells. The patient's own cells. We are utilizing off-the-shelf cells created by human-derived cord blood. So for that reason, that we will not have that issue that the FDA is warning about.
It's a fascinating technology you guys are in. So many solutions you're finding. Can you give us just a few closing remarks for our viewers? You've had a great presentation today.
Sure, Anna, thanks. I think it's a very exciting area. I know biotech's been roughed up over the last so many years, but I do think in general, it's starting to show signs of on its comeback, and biotech in general is always proved to be one of the best areas to be invested in as the markets return. And I do think Coeptis is set up very well to appreciate during this time. I mean, one thing we have going for us is we have a great science team, but we also have our science that is not having any of the failures that you have seen through basically mostly all of our competitors. So our science is pushing forward, driving forward very well right now. And we're very, very excited about it. So I think we're a good company to take a look at. Thank you.
Good point. Good competitive edge. All right, Dave, thank you so much for joining us on Emerging Growth. We hope to see you again in the future with some more updates.
Thanks, Anna.
All right, everyone. Thanks for watching. We'll be right back with our next presenter.